everyone, welcome back to my YouTube channel. My name is Serena and I'm a biology PhD student at UC Santa Barbara, and for today's video I'm taking you through my day in lab. I'm gonna just talk through what I'm doing and it's all sped up because otherwise this video would be like 2 hours long. But basically, first thing in the morning I'm checking the OD600 of my yeast culture that I had set up to grow overnight. So the OD600 tells you the relative density of yeast in your culture. So you can take this by using a spec. You can see the cuvette port is opening, so first you have to blank the spec, and then I didn't show it, but then I took the measurement of my yeast sample that was diluted 1 to 50. So then I back calculated to figure out how much of that original yeast sample I would need to set up a new culture with an OD600 of 0.15. Then this is going to grow in the 30-degree incubator for about 6 hours. So in the meantime, I sit down at my desk, and in the morning I pretty much just check emails, make a plan for the day, and then in this particular day I had a lot of grading to do. So then I went to the cell culture room so that I could get something going. I needed to coat the wells of a 96 glass bottom well plate with fibronectin so that later on when I add cells they can adhere better to the bottom of the plate. And this is for imaging purposes because I'm planning to image some cells on Monday, and this day was a Friday. So I'm just doing that, and then once it's been coated, I'll come back to the TC room and do the bulk of my cell culture work. Okay, so it is a bit later, and I've been working on grading and doing some other stuff, responding to emails, etc. And now I'm going to go back into the tissue culture room and do all of my cell maintenance. So let's go. Okay, so we're back in the cell culture room and I'm gloving up, ethanoling the hood, and the first thing I always do is check the cells under the microscope. You can see that these ones are pretty much 100% confluent. So I just want to get an idea of what my cells are looking like before I do anything to them. And then I just bring the cells, I put everything in the hood, get everything I need together, and there's a lot happening in the next few minutes, but it's very repetitive. So basically the first thing I do is aspirate the media out of each of the flasks and then add some trypsin. And the trypsin will help lift the cells off the bottom of the plate. So as you can see, I'm just going through and adding trypsin to each flask. Say this town is for the ghosts. And I'm buried in the rocks. And then once the trypsin's added, you put the flasks back into the 37-degree incubator so that the trypsin can work better at lifting the cells off of the plate. And for this particular type of cell, it only takes maybe a minute, but I typically will leave them in there for anywhere from 1 minute to 3 minutes, just as I'm getting other stuff ready in the meantime. So I put them in the incubator, and in the meantime I'm just labeling some conical tubes because a couple of the cell lines I need to spin them down. The rest I'm just passaging, so I don't need to put them into tubes. So the 2 cell lines that I'm spinning down, I got out of the incubator, and I'm neutralizing the trypsin with some media. So I just add the media first, and then I start to, I think it's called titrate. So you basically pipette up and down, and you wash, using the media, you wash the bottom of the flask to lift any cells that were not lifted during the incubation period. And then I take the full mixture and put it into the conical tube so that I can spin them down in a centrifuge. And the purpose of spinning down the cells is so that you can separate the cells from the media, and then you can remove the media, which contains some certain amount of trypsin. And the trypsin is toxic to the cells, so by spinning them and then removing the supernatant, you are essentially washing the cells and removing that toxin, which is good in certain scenarios, like if you're using the cells for an experiment, which is what I'm doing. So I just put them in the centrifuge, and then I got the rest of the flasks out that I just need to do like routine maintenance on. And I'm just doing the same exact thing to these cells. And I just skipped forward because it gets really repetitive, but essentially just adding media, washing the cells. For these ones, I'm just throwing the cells away and then putting back in some fresh media so that any trace amounts that were left on the bottom of the plate will come up. And I was just showing you the cell pellet from the cells that I centrifuged, and I am resuspending the cell pellet. So I'm removing the supernatant and resuspending the cell pellet in fresh media, and then putting some amount on to the 96 well plate and then into a 6 well plate. So I have a couple different experiments going, so I just knew that I needed to plate some in the 96 well plate and some in the 6 well plate. And all the amounts that I'm plating, I kind of figured out beforehand, so I have an idea of what I need to do. Okay, so my phone is on the brink of death, I think, because filming all of that TC stuff was like 30 minutes of footage. But I'm going to film the last little bit of cell culture stuff that I have to do today, which is freezing some cell lines. So these are what the cryo vials look like, and I'm just setting out however many I need, labeling them. I didn't show it, but I made some freeze media, and then I took some cell suspension, mixed it with the freeze media, and then this is going into each of the cryo vials. So 1 ml per cryo vial. Then I'll put them into the -80 freezer for 24 hours before moving it to liquid nitrogen. So I just had an advising meeting with an undergraduate that I mentor, and now it is, oh, my watch is dead, 4:06 p.m. and nobody's here, everybody's gone home. So I'm starting my yeast experiments. I'm using a new protocol, so I'm just kind of sitting here reading over it and making sure I understand everything I need to do, and I'm spinning the cells down while I do that. So, yeah. So actually, while my cells are spinning, I think I'm going to go and get some ice, because I will need this at one point in the experiment. Okay, so at this point it's like 4:15 p.m. and I'm getting ready to start my yeast transformation. So the reason why it's so late, why I'm starting, is because the yeast, if you remember in the morning I set up a new culture, they have to do an outgrowth for about 4 to 6 hours so that they're in the logarithmic growth phase. So it's just really important that the yeast are in that phase when you do the transformation, because it helps them take up the DNA better. So a transformation is where you alter the gene expression in some sort of cell, whether it's a yeast cell, bacteria cell, mammalian cell, by getting it to take up DNA from its surroundings. So that is what I'm doing today with yeast. And in general, after I took the yeast out of the incubator, I spun them down. I had showed you the cell pellet, removed the supernatant, resuspended the cells in water, and then right now they are centrifuging, they're getting spun down again, and that's just a wash step. And then in the meantime, I'm setting up tubes, I'm making a master mix of a bunch of reagents, got my cells from the centrifuge, there's the pellet, and so I made the master mix with a bunch of different things that will help the transformation, and then I'm removing the supernatant, and I'm going to resuspend the cells in some volume of water that is specific to the experiment. And then after that, I'm going to aliquot that cell suspension into however many tubes I need for the transformation. So in this case, I'm only doing 2 different transformations. And then from there, I'm going to spin the cells down again. There's the pellet. And remove the supernatant in every last drop. And then I'm going to add a certain volume of the master mix that I had made before. So that master mix contains polyethylene glycol, lithium acetate, and carrier DNA, salmon sperm DNA. And yeah, so I'm adding that to each of the tubes, and then I'm adding the specific plasmids that I'm trying to transform the yeast with directly to their respective epitubes, as well as some water. And then I vortex the tubes in several pulses so that the cells can get mixed with the solution. And then I bring it over to our 42-degree heat block and put the tubes in there, and they will incubate for 1 hour. So I would say that this is what a typical day in lab looks like for me. I did spend more time than usual on like grading and stuff, just because it's the end of finals week and grades are due soon, so I had to do that. But typically during my downtime, I'm like updating my benchling lab notebook page, planning out future experiments, reading papers, doing more logistical stuff like ordering things that I'm going to need for my experiments, or if things are running out. I have an hour right now, well, less than an hour, maybe like 45 minutes of incubation, and then I'll be able to plate my yeast. So I have my plates there around the flame, just like drying out and coming to room temperature. I'll probably film more of these lab days in my life in the future, so you guys should let me know if there's like anything specific you want to see, or if there's anything specific that you're curious about in terms of like lab work. Maybe I can like do a little Q&A portion. Typically it's pretty hard to film in lab because other people are here and I don't want to film in front of other people, but today nobody was really here. You know, people were in and out. I just kind of decided to film this spur of the moment, so I didn't show my morning routine or anything, but if you go back to just like PhD week in my life, you can see my morning routine. So before the 1 hour heat shock is up, I use that time to label my plates, and that just saves a lot of time. And then I went and retrieved the tubes from the heat block and spun them down. And so now I'm just resuspending them in a small volume of water, and I'm going to plate 50 microliters of each cell suspension. So to do that, I just add the amount to the plate, and then I flame off the glass spreader so that it is sterile. Wait for it to cool a little bit, and then I just spread the cell suspension right into the plate. And it's important that you try to spread it evenly and that it like kind of soaks into the plate. So this part's kind of tedious, but it's not difficult. I wouldn't say that anything I do is really that difficult. It's more just like tedious, and you have to make sure that you're using sterile technique. And just be careful of like certain details, but it comes with practice, I guess. Yeah, so I'm just going through and adding the cells to each respective plate. And hopefully these will grow within a few days. And then I'm just parafilming all of the plates so that the cells don't dry out on them. And then finally I'm putting them into the 30-degree incubator where they will grow for a few days and hopefully some colonies will form. And then I just clean up my bench at the end of the day, ethanol everything, and yeah, it's a nice way to leave my bench. Okay, so my work day is finally done. It is 5:47. I hope you guys enjoyed this video, and thank you so much for watching, and I will see you in my next video. Bye!